In 2017, he joined the PhD program in molecular biosciences, and his first year advisor was Bonnie Firestein. And I need to publicly thank Bonnie because Cade asked Bonnie for suggestions about genetics labs with whom he should rotate, and Bonnie directed him to me. So thank you, because I think I won. So Cade joined the lab in 2018. He worked really closely with Bob O'Hagan, Dr. Bob O'Hagan, who's in the lab, in the room. So excited. Bob is now working at the NIH. Bob had identified a gene that's really important for ciliary stability, and that when it's mutated, it causes ciliary degeneration, and in mice and humans, it causes neurodegeneration. So he screened for suppressors of this neurodegeneration and identified all these mutations. And then this is where Cade steps in. So Cade focused on a mutation in one of these suppressors, and it was in a gene that encodes a kinase. So Cade is an exceptional microscopist, and he sees things like no one else does. So along those lines, he went to the marine biology lab at Woods Hole in, I don't remember what year, 2019, to learn advanced imaging. He came back to the lab and told us that we were doing something wrong for 18 years. It's like, oh my gosh.
It was just the cover slip.
The cover slips, but we didn't know that. And he published his first author paper, it was Power and O'Hagan in 2020. And then the pandemic hit. So that was really a huge deal for, as most of you know, for research labs, because we were basically shut down for a year. Cade and we met weekly. It was a trying time for all of us. Cade worked through an awful lot of stuff and made some remarkable discoveries. So, my lab, for 23 years, we study this part of the cell called the cilium, and we kind of ignore everything else. There's no other organelles. And what Cade found was that this kinase actually isn't inside the cilium. It is on mitochondria. So, which someone pointed out is appropriate, that his last name is Power and mitochondria is the powerhouse of the cell, which I love. So Cade has actually been the complete driver of his project pretty much from 2021 on. He got a lot of help from Chris Rongo and colleagues. Chris couldn't be here today. He earned, he wrote and earned an NIH pre-doctoral fellowship. These are hard to come by, so that's a great accomplishment. And I was talking to Cade's parents before we started, and what Cade's dad said was, Cade always asks questions, right? So, and that's the way he's in the lab. He is extremely intelligent. He has a creative mind. He thinks about problems in unique ways. He reads extensively. So I didn't know anything about mitochondria, and he came back and he was, hey, this is how we're gonna address this. We're gonna do paraquat, and we're gonna do that and this. I'm like, okay. And that's really a true sign of someone who's doing doctoral research and who's going to be a doctor himself. So without further ado,
Thanks, Marty. So my name is Keaton Power, and I will tell you today about my thesis research titled Uncovering the Ciliary and Mitochondrial Functions of the NEMA-related kinase NECL4. And as far as questions, I divided up my presentation, and I'll be taking questions after part 1 and also at the end. So save questions for then if you have any. So an outline of my presentation. I'll start with some background and introduction slides. Part 1 is NECL4 and its functions in cilia. Part 2 is NECL4 and its functions in mitochondria. And finally, my conclusions. So to start out, Cilia are an essential organelle that most cell types in the body have, even cell types you might not think of. And they have a huge range of functions, from sensing to shedding viruses and vesicles. For example, this is electron microscopy of lung airway cilia, and colored in pink are COVID-19 viruses that are being shed by the cilia of the airway. So they have signaling functions. Some of them move. For example, in sperm flagella, it's close to cilia. In the eyes, kidneys, almost every cell type you can think of. So because of that, there are a lot of diseases of the cilia, and those are called ciliopathies. And like I mentioned, cilia are on most cells in the body, so that means ciliopathies can affect basically any organ system or just the whole body in general, or can cause developmental defects. Most of the bar lab studies kidney ciliopathies. My focus has been in ciliopathies that can occur in the brain, and in specific, it's not typically thought of as such, but neurodegeneration can, in some cases, be classed as a disease of the cilia. So there's a lot of functions actually that neuronal cilia have in the brain, which is why neurodegeneration can be called a ciliopathy. And in particular, my research is focused on the areas of neuroprotection to prevent neurodegeneration, as well as changes in the cilia in aging and disease. So in order to study things like neurodegeneration, we have to use disease modeling and model systems, because it's really hard to study these kind of things in human patients. So for example, one of these cilia-related neurodegenerative diseases is child onset neurodegeneration, and these are sample brain scans, in particular causing loss of the cerebellum. And there's a mouse model called PCD mouse to study this same phenomenon. And in this mouse model, they lose a bunch of Purkinje neurons, which are a large part of the cerebellum. So disease modeling is essential for studying areas of neurodegeneration. So in the bar lab, we use my favorite model organism, C. elegans. And it's really nice for disease modeling because a lot of human proteins have C. elegans orthologs that are pretty well conserved. And they can also display certain phenotypes related to different human diseases. And there's also a really big range of genetic experiments you can do. So we model diseases such as neurodegeneration in the worm, and the hope is that from our research it can be translated to other model organisms, including mammalian models, and eventually possibly to human disease as kind of the ultimate end goal. So in C. elegans, the only ciliated cells are neurons. There are a couple different types of neurons located in the head and the tail of the worm, and cilia morphology can range pretty widely between the different cell types. The cells that I will be focusing on in my research are called the amphid neurons and the phasmid neurons. And these are a cross-section of these specific cilia in the amphid and phasmid channels. So getting into the research, both cilia and cells, specifically neurons, have a very specific cytoskeleton ultrastructure that has to be maintained. My focus has been on the component of the cytoskeleton called microtubules. They're shown in this really nice image in blue. And the dynamics of these microtubules have to be maintained by various proteins that attach to microtubules and post-translational modifications, which are proteins and other modifications added to these tubules after they're made. And these affect things such as transport of organelles like mitochondria and different proteins throughout the cell, which is really important for neurons since they have really, really long processes. So making sure energy supply and proteins can get to and from synapses is really important for preventing neurodegeneration. And cilia in particular, they have a very specific microtubule ultrastructure. So in C. elegans cilia, the microtubule structure is called the axoneme. In the amphibid phasmid neurons, there is a segment called the middle segment or doublet segment, and it has 9 doublet microtubules that are symmetrical around the edge of the cilium and singlet microtubules in the middle. And the distal segment does not have these doublets, it's only singlets. For most of my research, I'll be focusing on this middle segment with the doublets. And this cilium is separated from the rest of the neuron by this gate called the transition zone that stops a lot of things from getting into the cilium, such as mitochondria. So I mentioned post-translational modifications before. The post-translational modification I'll talk about in particular is called polyglutamylation or glutamylation. So this modification of microtubules occurs specifically on the B tubule part of microtubule doublets. So each of these doublets in the middle segment has an A and a B tubule that make them up, and glutamylation is thought to occur just on this B tubule. And it's just one of many modifications that can happen with tubulin, but I will only be focusing on glutamylation, which in mammals is accomplished by a whole class of enzymes called CCPs that remove glutamates from tubulin, and TTLs, which add glutamates to tubulin. And they, this whole process and all these enzymes are very tightly regulated to preserve microtubule integrity. And this is really important because it has been shown that dysregulation of this glutamylation and deglutamylation balance does result in early onset neurodegeneration in the PCD mouse model I mentioned earlier, as well as in human patients. So what happens, and this occurs in these Purkinje neurons in the cerebellum that I mentioned previously. So microtubules that provide tracks for the transport of organelles and proteins throughout the cells, they're subject to glutamylation regulation as well as other post-translational modifications. And when one or both deglutamylase enzymes, I'm particularly talking about CCP1, is lost, then microtubules in the neuron become hyperglutamylated, and through mechanisms that are still being studied, this causes neurodegeneration, and it's thought to be via dysregulated transport of mitochondria throughout the cell, because they're really important to be at the correct places, and if you don't have energy supply, then neurons can degenerate. So this kind of mechanism is what my thesis research has been largely focused on, because it's we have a really nice model in C. elegans to study it. So what I've been doing is kind of using the C. elegans cilia as a model to study the axon degenerate or hyperglutamylation that I mentioned in the previous slide, because the cilia are where we are sure in the worm that this post-translational modification happens. So in C. elegans, there's a homologous gene called CCPP1, which was originally characterized by Hagen. So what I'm showing here is another cross-section of the amphid cilia. And the section of electron microscopy that I'm showing is through the middle segment with doublets. So in a wild type amphid channel, they have 10 cilia, and each cilium has 9 microtubule doublets, like I mentioned previously, and a variable number of singlets in the middle. And in CCPP1 deglutamylase mutants, they sometimes lack some cilia. There's ciliary fragments or vesicles in the amphid channel. And what I am going to focus on is the fact that a lot of times the microtubule doublets are broken, specifically the B-tubule part of the doublets, which, as I mentioned, is predicted to be the site of glutamylation. So one way that we can use to study defects in ciliary structure, like those caused by CCPP1 mutation, is by an assay that I'll be talking about a lot called dye filling. So this just entails incubating worms with fluorescent dye, and worms with intact amphid and phasmid cilia become stained in the amphid and phasmid neurons by this dye, as shown in wild type. And in CCPP1 mutants, as the worms age, they become progressively dye-filling defective, or DIFE. And by adulthood, they're almost completely dye-filling defective. And this is just showing higher resolution of what these phenotypes look like. So normal or non-DIFE amphids and phasmids fill pretty bright with this dye. There's another phenotype called partial DIFE, in which some neurons fill with dye or they don't fill as bright. And then the DIFE, dye-filling defective phenotype, where they don't stain at all, which is what we see in CCPP1. So this assay with this phenotype was used in the screen that Maureen talked about. It was a genetic screen to find interactors with CCPP1 or contributing genes involved in the process of neurodegeneration or protection from neurodegeneration. So what happened, this is a forward genetic screen, so CCPP1 worms were mutagenized, causing random mutations throughout the genome. And the second generation of progeny was screened with this dye-filling assay to find suppressors of the CCPP1 dye-filling phenotype. So basically additional mutations that made the worms able to dye-fill again. And this is where NECL4 was isolated. And this is, this kind of screen it's really nice that we can do this in C. elegans, and it's still relevant to humans because a lot of these genes do have human orthologs. And some of them might be lethal in a mammal model, but non-lethal in C. elegans. So another reason why this model is really great. So NECL4, it's a NEMA-related kinase, which are typically involved in cell cycle. NECL4 doesn't seem to be, but I'm not sure why. So it has not that many functional domains. It has its kinase domain and armadillo repeats, which are probably involved in protein interactions, as well as this called the pest domain. And what that is a signal to the proteasome to degrade the protein. And it's found in a lot of proteins that are pretty tightly regulated temporally and expression level wise. And the homologue in mammals is called NEC10, and it has pretty much the same functional domains as NECL4, and it's pretty well conserved, especially in the kinase domain. And in humans, NEC10 is known definitely to be involved in ciliopathies. In particular, ciliopathies of the airway. So this is images of airway cell culture with NEC10 mutant that has the same mutation that was found in a human patient. And they find that there are defects with clearing of mucus from the lungs in these patients, and cilia morphology is also really defective. Additionally, NEC10 has also been linked to a disease called primary ciliary dyskinesia, which is essentially when the cilia in the body that are supposed to move, like in the airway, can't move properly. And this causes similar phenotypes in the lungs where they're not able to clear fluid and mucus out of the lungs, as well as other phenotypes in other cells. So NECL4, interestingly, its expression is exclusive to the ciliated sensory neurons, but what's also interesting, I think, is that it's not found in the cilia themselves. So it's likely to be involved in some kind of ciliary process, but not by being in the cilia itself. And these are the amphid and phasmid cilia shown in yellow, and NECL4 is shown in red, and it is not found in the cilium itself. It stays confined to the dendrite as well as the other parts of the cell that I'm not focusing on. And as Maureen mentioned before, it is specifically localized to another organelle, but not the cilium. So this is dye filling assay data, and I'll be showing a couple of these. So we separate the scoring by amphids and phasmids because there can be differences between the neurons in the head and the tail. So wild type worms are almost totally non-dye or not dye filling defective in white. In gray is partial dye filling defective, and black is dye filling defective. So in adults, wild type are largely normal. CCPP1 is almost totally dye filling defective. And here I'm showing two NECL4 mutants, one that came from the screen and one that is a deletion mutant that Bob caught separately to make sure that NECL4 is the gene causing suppression, and both of them significantly suppress the CCPP1 dye filling phenotype. But on their own, neither NECL4 mutant causes a dye filling defect on its own, so we know it's not involved in like ciliogenesis, which is the creation of cilia, but it is involved somehow, otherwise we wouldn't see this suppression. And interestingly, when we add too much NECL4 via overexpression on a plasmid, that overexpression causes a diaphyling phenotype on its own. So we kind of concluded that NECL4 destabilizes cilia, and I'll expand more on that later. So since we found NECL4 in a suppressor screen of a D-glutamylase mutant, we wanted to see if possibly the suppression mechanism was through modifying glutamylation. So to look at that, we had to use a fluorescent antibody which recognizes polyglutamylation. And that antibody stains in the cilia, in the head and tail. We'll be looking at the head. And I'll be focusing on the amphid cilia middle segment, which are these. So in a wild type with this antibody, we see the other cilia of the head, which I won't focus on. But the amphid cilia middle segment look like two kind of triangular bundles. So in CCPP1 mutants, we don't see this staining anymore, presumably because the cilia are degraded, so they're not staining. In NECL4 mutants, we didn't see any difference between the mutant and wild type. And surprisingly, we also didn't see NECL4 suppress the CCPP1 phenotype of the loss of glutamylation staining in the amphids. So we concluded that NECL4 kinase suppresses CCPP1 diaphyling defect through a mechanism that doesn't involve glutamylation regulation. So this is the model that we proposed in a 2020 paper, in which within the cilium there's regulation of post-translational modifications, including glutamylation, that is that's performed by opposing processes of glutamylases and deglutamylases, which glutamylation is predicted to negatively regulate ciliary stability in some organisms at least. And outside of the cilium, there's kinases such as NECL4 that also negatively regulate ciliary stability. But the mechanism and function of this cilia phenotype is unclear. So this is where I kind of focused for the remainder of my research. So one of the things I wanted to do was see if it's the kinase activity of NECL4 in specific that is what causes suppression of CCPP1 neurodegeneration, instead of like loss of certain portions of the protein, since the other mutations you lose parts of the protein. So I generated a CRISPR directed mutation of the predicted active site of NECL4, and I changed it to a different amino acid, and I called this allele NECL4 KD for kinase dead. And when we did protein modeling of this mutant version of the protein, it's not predicted to change the protein structure as a whole, really. It kind of only changes the active site and the pocket surrounding it, the binding pocket. So we kind of predict that the kinase activity is the only thing defective about the protein. So that's what's causing any phenotypes that we see. So I wanted to test diaphyling suppression with this NECL4 allele, and I found that this is similar to what I showed before with wild type CCPP1, and I found that this kinase dead NECL4 allele suppresses CCPP1 diaphyling to a similar extent, at least in the amphids, the phasmids are a little different. And on its own, it is actually less dye-filling defective than wild type after a couple days of adulthood. And in addition, this protection from ciliary degeneration lasts further into aging. So even as wild type and CCPP1 animals lose more dye-filling ability, with age, this suppression is still present. So this loss of the kinase activity protects even through aging. Additionally, I looked at localization of wild type and the kinase dead NECL4 protein with another CRISPR tag, neon green. As I mentioned before, I'm showing here where the phasmid cilia are located. As I mentioned, NECL4 protein only localizes to the dendrites and other parts of the neuron. They're not in cilia. And this is also true if I look at it in a CCPP1 mutant background. And what's really interesting is that the kinase dead version, which is also tagged with the same fluorophore, it doesn't localize to cilia, but in a CCPP1 mutant background, it does localize to cilia. So my conclusion from that is that this kinase dead version of the protein possibly has different interactions with tubulin that has dysregulated glutamylation. And this also is the case in another mutant called TTLL11, which is a glutamylase, and TTLL11 mutants we found previously don't have any GT335 staining, so we predict that they lack glutamylation. So NECL4 localization is the same in this mutant, except the kinase dead NECL4, it localizes to the distal parts of cilia in TTLL11 mutants, as well as double mutants of CCPP1 and TTLL11. So I thought this was a really cool phenotype. I'm not 100% sure of its significance, but I think it's really interesting. I also looked at glutamylation again using the GT335 antibody to see if it also did not suppress the loss of staining that we saw with the other experiment. So again, I'm showing the phasmid cilia. This antibody should stain the middle segment as seen in wild type. And in green, this is the same CRISPR NECL4 tag. So in CCPP1 phasmids, we don't see the staining anymore. In NECL4 kinase dead mutants, the staining looks normal. And then in NECL4 kinase dead CCPP1 mutants, the staining is not restored. So it's the same phenotype that we see in with the other NECL4 alleles. But in this image, you can also see the ciliary localization of NECL4 kinase dead, but not the staining. So we wanted to see what is happening with the ciliary ultrastructure of these mutants. If the microtubules look any better in the CCPP1 NECL4 double mutant, if they look wild type. So we performed electron microscopy on these mutants as well. So this is the same image that I showed previously with wild type and CCPP1 mutant amphid channel cilia with the broken microtubule doublets. So when we look at NECL4 kinase dead and double mutants, we see that in the NECL4 mutant on its own, the cilia look mostly like wild type. They typically have 9 microtubule doublets, some singlets in the middle, and we don't really see broken microtubule doublets. And as well in the CCPP1 NECL4 double mutant, we see for most cilia, they have 9 doublets that are symmetrical. Very few broken B tubules, which is significantly different than CCPP1 on its own. So we think that this is likely the mechanism by which Neckel-4 suppresses the ciliary degeneration of CCPP1 by stabilizing B tubules and microtubule doublets. And this is just an extra interesting thing we found in this double mutant. We sometimes see microtubule defects that are not seen in many other mutants, such as singlet microtubules around the edge where the doublets should be, in addition to the 9 doublets, and also additional doublets in the middle of cilia. So we're not really sure what the functional significance might be, but I think those are pretty interesting phenotypes. So I mentioned before that overexpression of Neckel-4 causes dye-filling defects, which I repeated here. And in addition, another lab found that they did a similar experiment and found that overexpression of Neckel-4 causes dye-filling defects and also causes shortening of the cilia. So I wanted to kind of build on this experiment and see if I could characterize it further. So what I did was I deleted this pest domain that I mentioned, which is a proteolytic degradation signal, which I hypothesized would cause overaccumulation of the protein and sort of an overexpression phenotype. And I call this mutant pest deletion mutant. It's also tagged with neon green, and it localizes similarly to wild type Neckel-4. And again, with protein modeling, it's not predicted to affect the overall folding of the protein besides loss of this small region of this pest signal. And I found that, interestingly, this mutant caused a progressive dye-filling defect, kind of similar to CCPP1 mutants, but not as severe. So in larval stages, they're partially dye-filling defective, and by the time they reach adulthood, the dye-filling defect is more severe. So the ciliary degeneration increases as the worm ages, similar to CCPP1. And when we look at this cilia ultrastructure of this mutant, this is the same wild type image I showed before. And when we look at the pest deletion mutant, we see some of the phenotypes seen in CCPP1, broken microtubule doublets, tubule asymmetry, and again, it's not as severe as the defect in CCPP1, but I think it does show that over expression or unregulated levels of Neckel-4 lead to this B tubule instability phenotype. So in summary of the last few slides, loss of Neckel-4 deletion or kinase dead increases B tubule stability and ciliary integrity, and too much Neckel-4 with the pest mutant or overexpression reverses that, decreases B tubule stability and ciliary integrity. So I used this to look at a couple candidate interactors of Neckel-4 to see if I could find any proteins that suppressed the pest deletion dye-filling phenotype. So in 2020, a paper was published using cell culture and human mutations to find potential substrates or targets of NEC10, the human version of Neckel-4. And they found a lot of potential targets that are involved in cilia-related processes. So I looked at these and found targets that were involved in transport of proteins in cilia, cilia length control, and kinesins, which are motors that move on microtubules. And I looked at these because these can apply to cilia that move and cilia that don't move. C. elegans don't have motile cilia, so I ignored proteins involved in only motile cilia. So with these proteins, I saw which had a C. elegans gene, and kind of wanted to see if any of these could be potential targets or substrates of Nectin-4. Testing that by dye filling. So I found that there wasn't a dye filling suppression by key 11, OSM5, or DIE5, which a lot of, or these are dye filling defective on their own, so that's not super surprising. But I did find interactions with the two kinesins that I looked at. So the kinesin CLIP4 suppressed the dye filling phenotype of the pest mutant significantly. CLIP4 isn't that well characterized in C. elegans. It's known to have a function of trafficking in the ventral nerve cord, but not really a ciliary function. So I was more interested in this kinesin CLIP13, which is known to depolymerize, aka destabilize microtubules in cilia. And this also suppressed the pest deletion mutant significantly, more than the CLIP4 mutant. So, so far with my data, I showed that CCP1 is involved in regulation of glutamylation, which is really important for ciliary stability. And Nectin-4 is also important for ciliary stability through potentially acting through ciliary kinesins such as CLIP13. And that's the end of part 1. Part 2 is shorter, don't worry. So I will take some questions now, but not too long.
So you looked at CCPP1 with the Nectin-4 kinase dead under the electron microscope. Have you looked at the other loss of function mutants of Nectin-4 with CCPP1 to see if they have the same EM phenotype, since there's like such a dramatic difference in the localization of your Nectin-4 KD versus your Nectin-4?
I don't think we ever looked at a double mutant with those Nectin-4 alleles. We have images of Nectin-4 deletion allele on its own, and it looks close to wild type, but we didn't get to do it in combination with CCPP1, unfortunately. Yeah.
Good forward. So I'm super interested on this age-dependent phenotype, the dye-filling defective phenotype. So actually, it's very common to many like disease-related mutation that kind of age-dependent progressive process. Do you have any kind of idea or comments or clue about why this CCPP1 phenotype is also kind of progressive?
My hypothesis would be that as the worm ages, the cilia kind of have to be maintained, because cilia are kind of, microtubules are dynamic, so like constantly being broken down and like remade. So my hypothesis would be that as the worm ages, all this regulation that has to happen kind of becomes less effective. Or that as the worm ages, this hyperglutamylation increases, like it accumulates because it doesn't have the CCPP1 enzyme to remove the glutamylation. So just during age, it just accumulates more and more, which leads to more microtubule instability with age. That would be, yes.
curious if you guys thought about or tried making the double mutant that lacks the pest domain and is kinase dead.
I have thought of that. I haven't done it, but the group that did this experiment with NECL4 overexpression, they also, I don't have it shown here, but they also did this with a different NECL4 kinase dead mutant, and it did not cause a difilling phenotype. So I would hypothesize that adding kinase dead and pest mutant would not be difilling effective.
Real simple question, so your NECL4 complete knockdown, your EMs there are fairly normal, but they have some defects, so I'm just curious, what are those defects?
For
Just the original knockdown, because you said even those
This
are
one?
not. Is that, I get the knockdown in kind of yeah, the one, there's no protein, that one is fairly normal?
Non-mutation.
No.
yeah, it looks, I didn't like extensively analyze those, but they look pretty close to wild type.
But that's what I wanted to get at. So what is the most sensitive part of this process? So when you say they're fairly close, what's abnormal about this?
It's hard to tell sometimes, because even in wild type, like if you see this cilium, at this part of the cilium at least, it's only singlets. Like occasionally in wild type, there's like some defects, like a broken microtubule or two. So I guess it's hard sometimes for me to say like this looks exactly like wild type, because even wild type looks like messed up sometimes.
Yeah,
I guess that's
wonder.
why I said close to wild type, because I'm not.
I just wondered if there's a true to like what's the most sensitive part when you change these microtubules. That was the first thing to go when you start doing this
A dumb question from a non-scientist. Is one of the end games here possibly to make any progress in degenerative diseases like Alzheimer's or Parkinson's?
Yeah, I mean, that's kind of the end goal of model organism research, to look at, like, you can do a lot more manipulation with model organisms, study, a bunch of different functions of proteins, and one day, like, translate, that research can get translated into mammals and then human diseases. So we use this to study like the fundamental processes that happen with neurodegeneration to hopefully be applied in the future to human-related research. So I will move to part 2 of my talk, which is shorter. This is about the function of NECL4 in mitochondria, which did not expect to find, but it's interesting nonetheless. So I talked before about CCP1 mammal neuron mutants that in the PCD model that causes neurodegeneration, and a mechanism that it's thought to happen through is the loss of mitochondria motility, which then causes neurodegeneration. So CCP1 has been shown in cell culture to be involved with mitochondria morphology, so their shape, as well as mitochondria motility. So this is neurons from the PCD mouse, and they showed that CCP1 mutant, the mitochondria are not able to effectively move from one part of the long axons to another, so causing neurodegeneration. And NEC10, the human NECL4, also has been shown to be mitochondria localized, so the overlap between NEC10 and mitochondria. And it's not shown here, but it has functions in mitochondria homeostasis and DNA integrity. So I thought NECL4 could be involved in mitochondria homeostasis or localization. So I made a fluorescent tag that goes to the outside of mitochondria called TOM20, outer membrane protein, and I found that NECL4 does localize almost exclusively to mitochondria, regardless of kinase activity. So the kinase dead mutant also is mito-localized. And this doesn't change in CCPP1 mutants. The mitochondria morphology changes, which I'll get to in a minute. And I also looked at this mutant called DRP1, in which mitochondria can't divide, so they become hyperfused. And I saw that the pattern of NECL4 changed to correspond with this defect. So the localization, I think, is pretty specific. And like I mentioned before, mitochondria do not go into the cilium. So this makes sense with NECL4 being dendrite localized. They're with mitochondria instead of in the cilia. And this is just higher resolution imaging of the same markers showing NECL4 on the outside of the mitochondria membrane. And this is just a time lapse of these markers showing that NECL4 is transported with mitochondria in the dendrites. So like I said before, both CCPP1 and NECL4 mutation influence mitochondria morphology. So here I'm showing the whole set of phasmid dendrites. In CCPP1 mutants, the mitochondria are fewer and they're a little bit rounder than wild type. And in NECL4, it's the opposite. There are more mitochondria and they're also longer. And in the double mutant, it's in between. And this is just quantification of those phenotypes, showing there are more mitochondria in NECL4 kinase dead mutants, and they're also longer. So I think NECL4 and CCPP1 kind of oppose each other in this type of regulation. Additionally, I did some functional experiments to see if CCPP1 or NECL4 affects mitochondria homeostasis. So one of these is to look at chronic oxidative stress, which is a common mitochondria stressor. So I wanted to look at if their lifespan is different when exposed to oxidative stress. So when I look at control conditions without oxidative stress, I see that CCPP1 mutants typically have a little bit longer lifespan, but not extremely different from wild type. But when exposed to a drug called paraquat, which induces oxidative stress, the lifespan of CCPP1 mutants is very extended. And this is a phenotype suppressed by NECL4. So this, I think, is saying that CCPP1 and NECL4 have roles in how the worms respond to oxidative stress as far as regulating lifespan, which is a process that is partly regulated by the ciliated neurons, but also with a lot of other systems in the body. So as well as the ciliary functions, there are some functions outside of the ciliated nervous system that both of these proteins have a role in, which I think is cool. And my last experiment I'll be showing also involves oxidative stress. I wanted to measure if CCPP1 or NECL4 mutant mitochondria have a higher baseline oxidative stress than wild type. So to do this, I looked at a fluorophore called Rhode GFP, which is excited at different wavelengths depending on if it's oxidated or not. And I take the ratio of these two emissions to determine relative oxidation state between mutants. And when I looked at CCPP1 and NECL4, these are the quantifications. It's not statistically significant for CCPP1, but it's a little higher than wild type. But NECL4 mutants are significantly more at a higher oxidation state than wild type, and a similar ratio is seen for NECL4 CCPP1 double mutants. So that's, I think NECL4 is downstream of CCPP1 as far as regulating baseline oxidative stress in the mitochondria in the ciliated neurons. So overall, I've updated my model to reflect the different types of experiments I've done. So like I mentioned before, CCPP1 and NECL4 involved in the regulation of ciliary stability, but they're also involved in regulation of mitochondria number and length, as well as regulating oxidative stress in the mitochondria of the ciliated neurons, but also involved in crosstalk between the cilia, nervous system, and the rest of the body. So what I can conclude is NECL4 negatively regulates ciliary stability by acting potentially through ciliary kinesins and by destabilizing B tubules in microtubule doublets. That it's localized to mitochondria and is involved in regulating morphology, localization, and homeostasis, as well as CCPP1. And my kind of hope is that this work can contribute to like a broader understanding of neurodegeneration and how it can be regulated by processes in several different organelles. And also that maybe one day NEC10 can represent a potential therapeutic target for ciliopathies and or neurodegeneration, which would be really cool. So, I want to acknowledge the bar lab, which I really love a lot. Everyone is amazing, past and present members. My thesis committee, of course, Maureen, Monica, Bonnie, and Mark. The whole Rutgers C. elegans community, but in particular a lot of members of Chris's lab. Chris couldn't be here, but he helped me a lot with the mitochondria experiments because he's the expert. My funding, the people at City University of New York that did collaborated with me for the protein modeling, and Dave Hall and Ken Nguyen that collaborated with for electron microscopy, and the support of my friends and family and my cat. And here I just have a bunch of my favorite pictures from the lab. This is a cross stitch that a former undergrad made. I love the bar lab because I do. I won't get too mushy, but I've had a really great experience in this lab. Like starting graduate school, I was kind of nervous because the kind of stereotype or cultural idea is that like grad school is terrible and make you like burn out and hate science and whatever, but like, my experience has been very much the opposite. Like it's supportive and amazing environment, and I want to thank everybody in the lab, especially Maureen, for creating this environment. And what I am doing after this, probably sometime in February, is I am moving to Boston to start a postdoc position in Max Hayman's lab at Harvard Medical School to study patterning of glia, which are support cells for the neurons. So I'm kind of working in the same sort of area as now, but that's fine because I like it. And my most important slide, there she is. That's it.
So I was struck by the localization of NECL4 on mitochondria, like it looks like it's in little domains of mitochondria.
Yeah.
Do you have any thoughts about that? And if you don't, I do.
Yeah, so one of the things that I think would be a cool future direction is to see if that localization corresponds to like any specific like mitochondria adapter proteins or kinesins or other mitochondria associated proteins. So for that we'd need the higher resolution, probably SIM. Yeah, that's what I would like to find out.
If I can say what I thought.
Yeah.
I was like, wow, that could be a mitochondrial like ER contact site or something like that. So
Yeah.
that would be cool too.
Cool. So, do we know the mitochondrial targeting sequence for NECL4?
There is not a predicted targeting sequence. I checked like a couple different sites where you can put it in and see if there's a predicted like domains and sequences, and I couldn't find one. And there's also no transmembrane domain, so I don't think it's like in the mitochondria membrane. And it doesn't seem to be imported, so I'm not sure how it's associated.
I wonder whether if you can dissociate the NECL4 from mitochondria, can you also see the same phenotype as the NECL4 mutation on the rescue of CCP1
Yeah.
diaphragm defect?
Yeah, what about the ones that are like able to enter the cilium, like in your KD mutant, like you see some of the intrusion into the cilium.
Yeah, NECL4 is there, mitochondria are not there.
Right.
So it's becomes detached at some point and enters the cilium. But yeah, other than that, I haven't found any mutants where it's not mitochondria associated, but I think that would be pretty cool. See if you can like uncouple that from the cilia phenotypes.
I think you said, didn't you, I'm sorry if you just said this, but the KD mutant not only goes to the wrong place, but it still stays at the mitochondria as well. It doesn't
Yes.
lose that.
Does it what?
It does not lose its, yeah.
No.
This might be a self-serving question, but I have a reason for it. Do you know if NECL4 and Aurora kinase A interact or regulate each other?
I don't know. I don't think it was a predicted interactor from the NEC10 study. So I don't know.
The reason why I'm asking is that there's evidence in somatic cells, and we've been trying to follow this up in oocytes of Aurora A localizing and regulating and it also sort of like in one paper has a mitochondrial localization signal, but I'm not really convinced about that, and I'm just wondering if there's like these centrosome proteins that are kinases that are also kind of moonlighting and having this other sort of function with mitochondria. Maybe there's somehow connected, but
Yeah, and like
just food for thought.
Yeah. Thanks.
What about the ARM motifs? Has anybody mutated them to see what it does?
No, so one of my alleles is an early stop that I think is before that motif, and it just looks like loss of function, but I don't think there's, like pretty much I'm the only one looking at this protein. I haven't made a like arm deletion. I think that would be a good future direction.
So any mammalian studies suggest any the kinase activity with nickel-4, whether it's dependent on binding to mitochondria?
Not that I know of. As far as I know, they've only looked at wild type localization, so that I don't know. Oh, one more?
Yeah, can you just go back one slide?
Just
Why is your cat wearing pizza?
because I thought it was cute. It's just a little toy for her to chase around, but she doesn't like it, so I just.
On that note, the final question from the chair of the genetics department. Thank you, Dr. Matisse. The committee will go across the way and grill Cade